So this is a chapter I put together. It's not really a chapter, it's just a virus PowerPoint, really. These little guys are particles, right? In each little circle is a polio virus. And they have like sort of like a hexagonal shape. It kind of looks like a beehive. It helps you see the shape. You know, and then the thing to keep in mind about viruses is that they're particles, meaning they're not cells. When you see this sort of structure, then it's all the proteins that make up or surround the DNA or RNA in the middle. It can be RNA as well. It depends, one or the other. Then they have what are called these like spike proteins. Right? They call it fiber there, but they're big, long spike proteins. And now some viruses, instead of just this protein coat around it, they're surrounded by a membrane. So there are two types of virus. They can be DNA or RNA virus. Either have one or the other. Then you have a virus that's naked without a membrane, or you can have viruses that have a membrane. Like COVID-19 has a membrane, which is good. And they have to get into a cell in order to make more virus, right? They can't make viruses on their own. This is a piece of DNA or RNA, and then this is a membrane. He's kind of showing it in brown. And then the green guys are the spike proteins. So in this depiction, you don't see this shape in the middle, right? You see more of that kind of thing happening, right? That's what this is. The red is the DNA or RNA, and then these blue guys are the proteins, but they're all inside. It's like a big vesicle, but the envelope is another name for the term membrane. So in the virus life cycle, the first thing a virus has to do is attach to a cell. And in order to do that, it uses a receptor. So receptors are proteins found on surfaces of cells that stick to things, right? Things stick to it. So a virus will use its spike protein to stick to or attach to a cell that it wants to infect. So if you have a liver, say hepatitis virus, right, which infects your liver, then it infects the liver cell, and it only infects liver cells because only liver cells have the receptor that hepatitis virus needs to get into the cell. That's what makes it specific. Once it attaches, then it wants to get inside the cell. Once it's inside the cell, it comes apart, and then you express or synthesize all the proteins of the virus. Then all those proteins come together to make the virus, and then the virus is released by whatever cell it infected. Host is the host cell, and then the spike protein attaches to a specific receptor. They're not showing it, the receptor, kind of the way I do on here. I think this drawing is a little bit better than this, but that's what's in your textbook, I think. And then this is, they're not showing the whole cell, they're just showing a small part, you know, say like this part of the membrane. So this is the cytoplasm. It sticks to a receptor, and then the particle gets into the cytoplasm, and then you want it to release its DNA or RNA, so that the DNA and RNA can be used to make the proteins of the virus, right? So you have attachment, and then penetration means that you get in. So this is another depiction of that. Right? The viral particle sticks to the host cell. It gets into the host cell, and the DNA from the virus is used to make the viral particles. Right? It makes what it needs. It's replicated. Then you turn all the parts of the virus to make the virus, and then the virus is released. There might be, like, say, 4 genes the virus has, and those 4 genes become 4 different proteins when you express the gene. Those 4 proteins are the proteins that have to be put together to make a virus. So the virus forces the cell that infects to make more virus. In the process, the cell that does that dies. So if you get the flu, right, it infects epithelial cells in your upper respiratory tract. Those cells die, and that's why you get a runny nose or a fever, you know, things like that. They're all dying, and luckily they get replaced, but while they're being infected, you're losing a lot of cells. You know, if you have hepatitis virus, then it's killing liver cells. And that's why you have the problems that you have, right? So you make the proteins, then the proteins are assembled. Once you've assembled the virus, then the virus gets released. So one cell that's infected may produce 100 or 200 viruses before it dies. HIV, we say it has the cell that it infects has what's called a CD4 receptor, right? That's what this, and then the cell, these are called T cells. Right? T cells are white blood cells that HIV infects, because this is HIV, right? It uses its spike protein here and attaches to, this is the receptor on the T cell. So you don't see the whole T cell. It's kind of like this would be the T cell down here, and then this would be the surface of it, and that's one of those receptors right there. GP41, the green guy, is a spike protein on HIV. So once it attaches to this T cell, then the virus can get inside the T cell and kill it. If you lose all your T cells, then you get, you know, it's called acquired immunodeficiency. That's what AIDS is. Right, your immune system doesn't work properly, and you have AIDS-related disease illnesses, all kinds of things. So you can get HIV through sexual contact or using dirty needles. Right? If there's some of the virus, it probably doesn't take a lot, maybe 10 or so virus particles. If they get into cells and then replicate. So if you take an AIDS test and they might say, okay, you're HIV positive, you can't donate blood. That's kind of how that happens sometimes. That's how you find out. That means that you were exposed to the virus. It doesn't mean you have AIDS or HIV. It means you may have had it in the past. Now, if you have an active case of HIV, meaning now it's replicated, then it can turn into this disease. And it usually means that you've killed so many T cells in your body that you no longer, the count gets below 200 is one way of looking at it for diagnosing. All right, so if you had an HIV positive test, that means you have the antibody, then they might check your T cell count, and they can do a PCR test like they do with COVID-19, and that would tell them if you have the virus itself in your bloodstream. So being HIV positive doesn't mean that you have AIDS or that you're making any of the virus. You can get over the virus. The same thing with hepatitis C. So these are illnesses that you get that if you have AIDS, your T cell count's so low that you start coming down with infections because your immune system isn't working properly. Immunodeficiency, right? It's kind of self-explanatory with that. And then hepatitis C, it's a big problem. People got it for lots of reasons. It's known to cause liver cancer. And you can get a positive test for hepatitis C by donating blood. That's looking for the antibody, just like with the HIV test. So there's like 3 or 4 tests that they test all blood products, right? If you donate blood, then you're going to get 4 or 5 tests that it's looking for. And most of the time, they're looking for antibodies to whatever they're screening. Because if you've had hepatitis C or HIV, you produce antibodies probably for the rest of your life. COVID-19, same thing. But hepatitis C is a big problem. So many people have it. Now, well, this is another way of determining if there's virus, if your body's producing the virus. Instead of using the antibody test, that's a quick, cheap way of screening the blood. After that's positive, you could do an RT-PCR, it's called. This is the test they use for COVID-19 as well. And if this test determines there's the RNA of the virus in your blood, then you've got an active case of HIV or hepatitis C. Okay, and the problem with COVID-19 is the test that they used for that, for 2 years, they stopped using it because it tested for other things. Because in Africa they have these fruit bats, and there's always the virus in these fruit bats, and it can occasionally get to other animals because of the bat. And if people eat these animals or the bats, then they can come down with Ebola, which fortunately for us, we don't have fruit bats in the United States, so we will never really have to worry about Ebola coming here. So endothelial cells, those type of cells are found in our blood vessels. If too many of them die, then the blood vessel gets leaky and you start bleeding from all sorts of places. And that's what can kill you within a couple days. The HPV is short for human papillomavirus, right? And this virus infects skin cells, keratinocytes, right? And then it can cause cancer of cervix, that's the main one, but there are other places too. So it's the skin in these areas, or the mucosal membranes in these areas, that get infected by the virus, and then those cells, because they were infected, become cancerous. So, right, if you get a vaccine, hopefully, to prevent you from getting this virus, then it could prevent you from getting cervical cancer. Because you don't get infected by, hopefully it protects you from the virus. I don't know if I believe in vaccines anymore. You know what I mean? I was never against vaccinations, but, you know, I'm wondering now, with what they've done recently, why anybody would trust them with anything like that. It's all because of this. So the coronavirus is a virus just like the flu, right? There are lots of them, right? There's the swine flu, the bird flu, right? They're basically the same virus that's somewhat different. They still infect the same cells in your body, but they might be more contagious or more deadly. Corona has the same reputation as the flu. And you know, when it first came out, we thought it was the end of the world, right? I mean, it was just ridiculous. SARS was an old name used for an outbreak. I think it was in the 2000, maybe even before 2000s. And then MERS came, I think, around in 2005. And then we had SARS-CoV-1 and then SARS-CoV-2. COVID-19 refers to the disease that SARS-CoV-2, the virus, causes. It's kind of like HIV causes AIDS. This is the disease, right, the name of the disease. But we always say COVID-19 refer to the virus because it's simpler. But it has a membrane, right, and then there's this coil here is the RNA, and they're kind of leaving off the protein that protects the RNA, and then they have these spike proteins, just like most viruses do, or all viruses have them.